Okay, good morning, everyone. First, I would like to thank Professor Mason and the organizers of this conference for inviting me. I'm happy to be here. So, today I'm going to talk about our recent work on the molecular classification of prostate cancer. As many of you may know that I'm coming from the Michigan Center for Translation Pathology. This is the center from which we first reported the discovery of the recurrent gene fusions in prostate cancer. That is the EDS family of transcription factors. So after I joined in this group, my work was focused on looking at the genomic rearrangements in the EDS negative prostate cancer, where we found an interesting gene fusion involving BRAF gene fusions, which I'm going to talk in detail about that. So when you look at the prostate cancer, unlike many other solid cancer, they lack actually distinguished histopathological subtypes that differ in the prognosis or treatment response. But the recent genomic analysis, which actually provided evidence that prostate cancer can be classified into distinct molecular subtypes. So I'm going to talk about some of the work that we did recently. And also, when we looked at the gene expression profiling, that did not tell us much. We are not able to actually strictly classify the prostate cancer into distinct molecular subtypes, although the gene expression microarray has been done extensively. But there is a recent study from Sloan Kettering Cancer Center from Dr. Charles Sayers' group. They did a very integrative analysis of gene expression, copy number analysis, and resequencing, and they suggested that maybe an alternate approach to look for the molecular classification of prostate cancer. But our research is mainly focused at the Michigan Center for Translation Pathology, looking at the chromosome rearrangements, and based on our analysis of the prostate cancer, we show that there is an existence of the molecular subtype of prostate cancer based on the chromosomal aberrations. So as you know that, when you look at any solid tumor genome, there are numerous chromosomal rearrangements. Because of high aneuploidy and complex rearrangements, you will find several type of aberrations like amplification, deletions, duplication, all those things. Among various aberrations, I think gene fusions are emerging as an important class of new mutations in solid cancer, but which was not discovered for a long time, more than about 50 years. We did not have a good understanding about the nature of the gene fusion in solid cancer. Because when you look at about 80% of the all known gene fusions in human neoplasia are reported from the hematological malignancies, from leukemia and lymphoma. But when you look at the death due to cancer, is about 80% of the cancer death is due to solid cancer, but we have only about 10% of the gene fusions are coming from the solid cancer. So it is mainly because not because that we don't have that many gene fusions, but it is because of the technical limitations and also the clonal heterogeneity in the solid tumor. We were not able to discover the gene fusion for a long time. So what it tells us is we really have to look at the solid tumor genome by entirely a new approach. So that's what that was taken in the year 2005. There's an interesting analysis called Cancer Outlier Profile Analysis called COPA, which completely integrate the gene expression analysis or the gene expression data from all solid cancers and different studies for each cancer type. It was completely integrated. And with that analysis, what they found was, particularly in prostate cancer, when you take all the studies, what they found was the ERG gene was actually found out as an outlier gene among all the studies. So interestingly, what they found was, so in order to quantitate the expression level, why this gene is actually highly expressed, in order to assess the expression level, initially the first round of PCR was done using primers from the exon 1 and 2 and from the last exon. Interestingly, what they found was there was no expression from the 5' end of the gene, and there was a high level expression at the 3' end. So based on that initial finding, and primers were designed for each and every exon by using the exon spanning primers, and when they profiled the entire gene, what is interesting was they found the exon 1 to 3, there was no expression at all, and from exon 4, all other exons were expressed at very high level. So which indicates that this gene is not expressed as a normal transcript. This indicated that this gene may be a fusion gene. So in order to confirm that, RACE PCR, what is called the rapid amplification of cDNA ends, the RACE PCR was performed by using primers from the region that is overexpressed, and when you amplify the 5' end, we were able to confirm this gene indeed was a fusion gene rather than a normal transcript, and the 5' partner was the TMPRSS2 gene that was actually identified as fused with that gene. And by using, after identifying the 5' gene, and fusion PCR primers were designed and the fusion transcript was confirmed. So in order to confirm that, this is not a PCR artifact, so then later, indeed, this DNA rearrangement was confirmed by using the fluorescent in situ hybridization, by using the break-apart FISH probes, and we confirmed that this fusion is produced either by deletion or by translocation. So this is the very first discovery. By using an entirely novel approach, we discovered a recurrent gene fusion in prostate cancer. So soon after that, with the same approach, by using the cancer meta-outlier profile analysis, after the discovery of the TMPRSS2 gene fusion, there is a second gene called the SPINK1, which was found to be overexpressed in a subset of prostate cancer. Interestingly, these cancers were negative for the known AIDS family gene rearrangements, which was confirmed about 10% of the prostate cancer. And interestingly, the expression of this SPINK1 can also be monitored in the urine for routine diagnosis purposes. So this was based on these two reports, which gave us the very first evidence of existence of the distinct molecular subtype of the prostate cancer. So as you can see, now we can see actually two distinct types: AIDS family rearranged set of prostate cancer, and another one is the SPINK1, which account for 10%. So soon after the discovery of the AIDS family gene in prostate cancer, there was actually extensive research was actually done across many labs all over the world, which confirmed the existence of the AIDS family genes rearranged in prostate cancer. Because there are 29 AIDS family genes, but in our lab, we did a very systematic screening for all the 29 genes by using the fluorescent in situ hybridization. What we found was either ERG, ETV1, ETV4, and ETV5. Only these are the 5 AIDS family genes that are known to be rearranged in prostate cancer. And interestingly, when you look at the AIDS family genes, the 5' partner genes are mostly, it's not all of them, mostly these are all androgen-regulated genes, okay? So all the AIDS family genes are fused with the androgen-regulated genes, which drives the overexpression of the AIDS family genes in these samples. So based on this data, we can actually classify the prostate cancer in two major groups, like AIDS family genes and the SPINK1. Then again, within the AIDS family, we can classify them into ERG, ETV1, ETV4, and ETV5. These are all the 5 distinct subtypes. So we asked the question, what happens to the remaining about 30 or 40% of the prostate cancer? They do not have this AIDS family gene fusion, okay? That's where we engaged in a study, because we knew that the cancer outlier profile analysis is not sufficient to identify all the molecular aberrations. So we undertook an entirely an unprecedented or a novel approach by using the next generation sequencing technology called the transcriptome profiling of AIDS negative prostate cancer. So, and also we know that all the AIDS family genes that are identified in prostate cancer, they are not druggable because of the transcription factor, because of the high homology across all the AIDS family genes, they are not druggable. So what we found at that time, there was a report that showed the EML4-ALK gene fusion was identified in a very small subset of non-small cell lung cancer, and this kinase was druggable. So this was the basis for our hypothesis that we could identify, although not a high percentage of the tumors, even actually a small percentage of the tumor can be identified with the druggable target gene fusions in solid cancer. So with that working hypothesis, we engaged in the next generation sequencing technology. Using that, we profiled the AIDS negative prostate cancer. So for those who are not familiar with this technology, what is next generation sequencing technology? I'm not going to spend too much time actually digressing what I'm talking. Just one slide to show that by using next generation technology, this is very simple. All we need is about 1 microgram of DNA from the tumor samples. Once you have the DNA, you have to do the polyase selection and make it double-stranded, make a cDNA, and you need to fragment the DNA. And after fragmentation, you need to size select them from either actually 300 or 400 base pair. You need to size select the fragments, then add the adapters at the end of each fragment. Then you attach this adapter into the flow cell. This is in the sequencing machine. They attach to the flow cell, then we start sequencing. So when you do the sequencing, because we actually pioneered in this approach, in the last 2 years we published these 3 papers in Nature, PNAS and Nature Biotechnology, and showing the utility of this approach to discover the gene fusion in prostate cancer. So how we do this sequencing? So once you have these fragments with all the adapters attached to the flow cell, you can do the sequencing by 2 approaches, which is called either single read or paired end approach. If you sequence only from one end of the fragment, it's called single read sequencing, or you can sequence from both ends of the fragment, it's called paired end sequencing. And you can sequence minimum about 36 base pairs and up to 100 base pairs. Because of the improvement in the technology, you can sequence up to 100 base pairs. So when you do the sequencing, you get the sequencing and you can actually, what we call as the mate pairs that you can generate from either end of the sequences. Okay? So once you get the mate pairs, then you can actually start mapping them to the known reference genome. So when the reads are coming from the normal transcript, they map actually perfectly to the expected genomic locations. And depending upon the number of reads, you can actually tell the quantitative expression of the genes. And what is happening is, interestingly, among all the mate pairs, you will find a set of reads that are not mappable to the reference genome. Because these ends of these fragments are coming from 2 different genes. So only from these we will be able to identify the gene fusions. So with that, what we did initially for a proof of concept, we used the VCAP cell line and we used the technology to see whether we can actually identify the TMPRSS2 ERG gene fusion. So interestingly, when we do the sequencing, we were able to identify several reads. And depending, because in VCAP, this TMPRSS2 ERG is expressed at a very high level, we found a lot of reads, and each read is unique. They are not exactly having the same start and same ends, but what we call it as actually each read is a unique read, and which shows the TMPRSS2 ERG gene fusion. So with that approach, we were able to sequence its negative prostate cancer. So one thing here I want to point out that, because as we know that the VCAP genome or any prostate cancer, you will find, because of the complex rearrangement, you will find many gene fusions. Okay, we are not going to, we did not identify not only the TMPRSS2 ERG in the VCAP cell line, we identified several gene fusions. So now the challenge is the informatic challenge. How do you identify among the various gene fusion, what is the driver gene fusion that is responsible for the cancer? So in order to do that, we sequenced even the K562 cell line in which the BCR-ABL gene fusion is the driver gene fusion. So what we found was, among the various gene fusion, interestingly, we found out that the driver fusions are always actually enriched in terms of actually the sequencing reads. Okay, so when you rank all the gene fusions based on the number of sequence reads that you identify from each samples, you will be able to tell which one is an important gene fusion for that particular sample. Because you may not find that particular gene as an outlier across the entire cancer, but as far as the individual sample is concerned, you may find that particular gene may be expressed at a very high level. So with that idea in mind, we sequenced the EDS negative prostate cancer. So even before doing that, until then we used only the prostate cancer cell lines for sequencing. So this for the first time to confirm whether we will be able to identify these driver gene fusions in primary tissues. So here what we found, we used 2 samples, one containing the ETB1 gene fusion, other one containing the ERG gene fusion. We sequenced 2 samples, and we were able to find out there are many gene fusions from individual samples, but we always found the ETB1 or the ERG, they're ranked among the top gene, so which actually give us more evidence to show that we will be able to identify the driver gene fusions. Okay, so with that idea, when we sequenced the EDS negative prostate cancer, we sequenced several samples, but interestingly, we found 2 samples. In one, we found the rearrangement with the BRAF gene, and the other one with the RAF1. So both the BRAF and RAF1 are the same family of kinase gene fusion. But interestingly, what we found, in one sample, we found SLC45A3 BRAF. In other one, we found both RAF1, ESRP1, and ESRP1 RAF1. So these are essentially the same gene fusion that is created by the balanced reciprocal translocation, and both rearranged chromosome is actually making a fusion transcript. Because which is very rare, which we don't find normally because of the power of the next generation sequencing technology, we were able to find out the reciprocal gene fusion as well. So then because of the biological importance of the BRAF gene, because we know that this gene is involved in a variety of cancer, especially in melanoma, about 50 to 70% of the melanoma patients are known to have mutations in this gene. But here we found rearrangement. So when we looked at the expression profile across many samples, both the normal and the PCA, among the many samples, the index sample is showing a very high level of expression of the BRAF. Okay, so then we engaged in a very systematic evaluation. Because of the sequence reads, we know that what is the exons that are fused in this gene. So here we found the first exon of the SLC45A3 is fused with the exon 8 of the BRAF. Okay, so based on that, we designed the exon spanning primers. When we did the quantitative PCR, what we found only after exon 8, the gene was highly expressed, and from exon 1 and 7, there was no expression at all. So which confirms the fusion. And also when we look at all the sequence reads, only from exon 8, you will find actually many reads actually supporting the expression, but there was no reads coming there. So with that, we were able to confirm the presence of this BRAF gene fusion in that particular sample. And secondly, we confirmed the same way we confirmed the RAF1 and the ESRP1 gene fusion in the same sample, because these two gene fusions are occurring in the same sample that is produced because of the balanced reciprocal translocation. So having identified this gene fusion based on the sequencing technology, we engaged in a very systematic validation of the gene fusion. So this is by the quantitative real-time PCR. We actually confirmed the presence of the gene fusion only on the index samples. Although we actually screened across many samples, we did not find the same gene fusion in other samples. This is mainly because because the 5' partner is not always the same, because of the promiscuity of the 5' partner genes. We cannot use the fusion PCR strategy to screen a large panel to identify the recurrence. Okay. So, and also, in order to confirm whether the gene fusion is produced by the DNA rearrangement, just make sure, since we are actually getting the gene fusion by transcriptome sequencing, we want to make sure that this is not a PCR or actually sequencing artifact. So we want to confirm this gene fusion is indeed created by a DNA rearrangement. We did a FISH analysis both by dual color split probe and also the fusion probe, which confirmed the rearrangement for both the genes. So this is for the BRAF and this is for the RAF1 gene fusion. So here we are showing the splitting or the rearrangement of the gene, and also we used the probe both from SLC45A3 and the BRAF, and we showed the fusion, and similarly we did the same thing for the RAF1 as well. And we did not stop there when we looked at the validation. So what is the gene fusion? So now we confirmed the gene fusion at the RNA level. So do they really make a protein? Okay, so when we looked at the transcript structure, indeed they form a perfect open reading frame, and when we did the Western blotting using the index sample, we were able to find the truncated BRAF that is expressed. We have a truncated protein, and also similarly the RAF1 also we confirmed the expression of the fusion protein in the index sample, as you can see the PCA17, because here this is because of the fusion, it created a large protein about 120 kilodalton, which is expressing only in the index sample but not in any other sample. And also we cloned the full length transcript of this gene fusion and we expressed in the HEC293 cells, and there also we actually confirmed the protein. So this for the first time we were able to validate the discovery of the RAF gene fusion in prostate cancer, and we confirmed both at the RNA level, DNA level, and also at the protein level. Okay, so having identified this important biologically important gene fusion, next we engaged. Before that I want to show you that we wanted to see, since it is involved in the prostate cancer, and we want to see whether the 5' partners that is fused with the BRAF and the RAF1, whether they are androgen regulated. So we found that only the SLC45A3 is an androgen regulated gene, but not the ESRP1 is not an androgen regulated gene. And so having validated this gene fusion on the index samples, so we want to actually screen a large cohort of the prostate cancer to see for recurrence of this gene fusion. So we screened about 349 samples, and out of which about 6 samples were known to have the BRAF rearrangement by FISH. And out of 450 samples, 4 samples are known to have a RAF1 rearrangement, so which confirms the recurrent nature of this gene fusion. And interestingly, when you look at all the samples except one, all other samples are negative for the X family rearrangements, which confirms that we found this gene fusion only in the X family rearrangement negative prostate cancer. And also about half of this prostate cancer are actually castration resistant. Samples which shows that and these gene fusion occurs in a very rare type of prostate cancer. So having established the recurrence and the confirmation of the BRAF, we wanted to study what is the relevance of the gene fusion in prostate cancer. What is it really doing it? Is it really causing cancer? So what we did, we engaged in a very systematic validation study to identify the oncogenic property of the gene fusion. So for that, what we did, we cloned the full-length gene and put them in the expression vectors, and we did a lot of these in vitro and in vivo studies. In this one, we checked for the foci formation of the SLC45A3 BRAF gene fusion. So indeed, they formed a very high frequency of the foci formation, which confirms oncogenic property. And also, we made the NIH3T3 cells and we put it in the nude mouse, and we confirmed the formation of the tumor that is actually induced by this fusion transcript. And we followed this tumor formation up to 5 weeks, which confirms that this fusion induce cancer. And also, interestingly, what we found when we look at all this fusion transcript by cell proliferation assay in prostate background. So far, we did only in NIH3T3 cells. Here, we actually introduced this fusion transcript in the RWP, that is a prostate cancer epithelial cells, and we found that there is an increase in cell proliferation. But interestingly, we were able to inhibit the induced cell proliferation by using the inhibitors that is specific for the RAF kinase. So in this experiment, we used the sorafenib, which is a drug, which is known to inhibit this kinase, and we were able to inhibit the induced proliferative activity of this gene fusion. And also we looked at the invasion of this transcript, both the SLC45A3 BRAF and the ESRB RAF1. They showed actually increased cell invasion, and you can actually inhibit this invasive property by using both the sorafenib and also MEK inhibitor, which is U0126. We were able to reverse the oncogenic property of this gene fusion. And also we confirmed this by Anchorage independent growth assay. By using the sorafenib and the MEK inhibitor, we are able to inhibit the oncogenic property of this gene fusion. And also, since these genes are acting in the MAPK pathway, and we check the phosphorylation of this gene. So in fact, these two gene fusions, they induced actually phosphorylations under expression conditions, and you can inhibit this phosphorylation by using the MEK inhibitors. Okay, and so this is for the first time, and we were able to show that the oncogenic property of the RAF gene, and which can be inhibited using a drug. Okay, then. So here, what is the significance of this RAF gene fusion rearrangement in prostate cancer? So this is for the first time we identified the non-ETS gene fusion identified in ETS gene rearrangement negative prostate cancer. And the BRAF and RAF1 rearrangement positive patients, interestingly, what we found was they do not harbor the canonical BRAF V600E mutation. So all these patients with the gene rearrangement, they do not have the mutations. As you know that, we have the PLX4032, that is the drug that inhibits only the mutant BRAF, and it will not work on this fusion, because we did not publish the data yet. We collaborated with Professor Neil Rosen, and they did an extensive study and showed that we cannot use that drug to treat these patients. Okay, so although rare, we identified a subset of prostate cancer identified in non-ETS and which is a druggable target gene fusions. And although BRAF mutation are almost non-existent in prostate cancer, this is from Dr. Linda Chin's work. They showed in mouse models it may cooperate with other mutations, it may cause cancer. And the ETS transcription factors are also, including ETB1, is shown to be downstream targets activated by the RAS-RAF pathway signaling, which suggests a possible common pathway. And we can actually develop new diagnostic assays based on PCR and FISH for screening patients with the RAF gene rearrangement, and these patients may benefit from the RAF kinase inhibitor. So based on this discovery, we are actually introducing a new classification of the prostate cancer. Now, in addition to the AIDS and the SPINK1, we have another class of what's called the RAF1, or 1 to 2% of the prostate cancer. So it is not only the discovery of the gene fusions. What is interesting is actually targeting drugs can be developed for these gene fusions, because I think early evidence show that the X family genes, they work in a DNA damage pathway, they induce a lot of DNA breaks, and we can actually try the PARP or the actual small molecule inhibitors targeting the X family. And anti-SPINK1 antibodies can be used to treat this group of patients with SPINK1, and we showed that RAF and MEK inhibitors can be used for the RAF. So all these molecular targets can be targeted with the actual potential drug that will help us to actually treat these prostate cancer patients. So I think this is time for the kind of individual medicine that actually, because all the patients, you cannot actually see them as actually same. Each one, you can classify them based on actually the molecular aberrations, and you can target them with individual drugs. So with that, we have a new kind of diagnostic protocol. How once you have a prostate cancer patients, how you can actually classify them. Initially, first you can do either by doing the morphological analysis, you can classify them whether they are cancer or benign. So then optionally you can check the AMA-CR and the cytokeratin and the P63 as a basal cell markers. So once you have the cancer, initially first you can actually do the ERG-IHC and the SPINK1 IHC. As actually some of you may be aware that there is a very good antibody that is available that is for ERG, which is very highly expressed only in the H positive samples, but not in the normal prostate epithelium, which has about 98 or 99% sensitivity and specificity. This paper was published in New Pleasure recently. I didn't have time to talk about that. So we don't have to do FISH initially. You can actually directly go with the ERG-IHC and the SPINK1 IHC. And if you have ERG positivity, you can do FISH, which is an optional, because there is a two different type of actually FISH rearrangement, whether it's a 5' deletion or split. As we know that 5' deletion, especially in metastatic prostate cancer, it goes to all the bone metastasis cases are known to have associated with a 5' deletion. So for that reason, you can actually include. And interestingly, so if you find the ERG and the SPINK1 is negative, then you can actually go to the FISH analysis of the ETV1. Then if that is negative, you can go to the ETV4 and ETV5. If you find that also negative, then go for the BRAF, because we ordered them based on the frequency of these aberrations, because these are all kind of rare, but still they can be investigated. If all of them are negative, then at the end you can do the AR, MAKE, and the P10 analysis. And even if you find everything is negative, and just save the samples and go for the next-gen sequencing, definitely you will find some interesting aberration. Last, only 2 slides, I want to show you that because of the significance of the BRAF. As you know that, it is not only we discovered the BRAF in prostate cancer, because we did a very extensive screening by using the tissue microarray by FISH screening. We screened melanoma, breast, lung, endometrium, and all different cancer. What we found interestingly, both in melanoma, prostate cancer, and in gastric cancer, we found the BRAF rearrangement in actually 1 to 2% of these samples. And for the first time, we showed the RAF1. Initially, it was reported to involved in brain cancer. We showed that involvement of the RAF1 in prostate cancer as well. So although then, if you look at the molecular structure of this fusion gene, although all different cancers, they may actually fuse with different 5' partners, but in all the cancers, it always retain the kinase domain, which is truncated, which is constitutively activated in all these cancers. So based on this, what we can find out is, although we found mutations are more prevalent in melanoma, but we found actually other than the mutation, rearrangement in rare cancer types have been reported, but not investigated for the oncogenic property that we showed very clearly that this rearranged RAF can also induce cancer. So the transforming activity of the fusion genes lacking the regulatory domains, BRAF, which results in the constitutive activation, and tested kinase inhibitors effectively reverts the oncogenic activity. So although rare, a distinct subtype of prostate cancer can be identified with the druggable gene fusion other than the H family genes. And screening for RAF rearrangement by FISH, PCR may be useful for identifying cancer patients who may benefit from RAF kinase inhibitors. And although it is rare, but low incidence in prostate, gastric cancer, melanoma, thyroid, and breast cancer, we found that. So the deep sequencing of cancer transcriptome analysis is an alternate approach to identify targetable gene fusions. So here I want to actually conclude by saying that identification of RAF gene rearrangement is actually in several solid cancer, irrespective of organ site, indicate a common molecular mechanism operating in the genesis of actually cancer. So that's why we have to look at the molecular aberrations rather than the histological aberrations in different cancer, which will help to actually profile the cancer based on the molecular aberrations. So with that, I would like to acknowledge, because this is actually a big team effort, I would like to acknowledge all the people who are involved in this study. First, especially I want to thank Dr. Arul Chennaiyan, who was supposed to be here to give this talk, but he deputed me to come here. But I want to thank everyone, and also, again, the organizers for giving me this opportunity. Thank you very much for your attention.
Perhaps I could open the session with what appears to be contrasts. On the one hand, Nalis, you have highlighted how using not an exclusion, but looking at gene outliers, you find the fusion gene. It's present in a small proportion of prostate cancer patients. And doing some sums in the back of a postcard, we might be looking at 5,000 patients in the US per year with a BRAF gene fusion, which will not make the drug companies happy. But you would predict these patients will be exquisitely sensitive to a BRAF inhibitor or a MEK inhibitor. On the other hand, Jan, you highlight a very important but almost ubiquitous gene mutation, which potentially does open the door for targeting maybe not the tumor suppressor gene itself, but for targeting the pathways that are interacting. The question that I would pose is, where are we going? Are we going down the route of a very individualized therapeutic targeted strategy, in which case do we need to open this out beyond prostate cancer? Or are we still using the blunderbuss approach for chemotherapy for patients with prostate cancer? Now, maybe you could comment.
Yeah, I think although the BRAF rearrangement is found in a very small percentage of prostate cancer, but when you look at the overall incidence of the prostate cancer, it will identify around like 3,000 to 5,000 cases. And other interesting part is, just to make the drug companies happy, it is not only found only in prostate cancer. We are seeing in other cancers as well, more than like at least 3 or 4 different cancers. So I think initially the important thing is, especially for each cancer, when we look at the histopathological examinations, it is hard to tell much what we can do, in order to actually select the patients for appropriate treatment. So I think this molecular classification is actually a new beginning, since we have entirely a new technology and new tools that are available to classify this cancer. Because we have a new tool to look these cancers in an unprecedented way, especially by sequencing all these cases. We will be able to identify a small subset of cancers with entirely a new molecular aberrations, which we would have not actually find out by any other conventional methods.
So is the BRAF gene mutation or the RAF gene fusion, is it one of a number of gene fusions that you're going to bring to the arena over the next number of years? So are they multiple? Are they common but different types?
Exactly, yeah. Because I think based on our initial analysis, we did not actually extend the analysis in a larger cohort. We initially, based on our finding from prostate cancer, we looked into at least 5 different solid cancer. It is not present in all the cancers. We looked at lung, breast, endometrium, liver, and other cancers. We did not find that gene fusion in other cancer. So we found only in prostate, melanoma, and gastric, and earlier reports they identified in brain cancer and thyroid cancer. So there has to be some restricted common molecular pathway in a small sets of cancer, that might identify a small, kind of
this
subset.
This question.
Simon Brewster from Oxford. Could I ask Professors Palanisamy and Trapman if your respective studies have included pin lesions, And if so, do your studies strengthen the status of PIN as a pre-malignant lesion, and could that also be targeted?
Yeah. So I think from our analysis, I think even the studies are ongoing in our lab itself. We haven't looked specifically for the BRAF aberrations in the PIN lesions, but we have evaluated, and there are reports, they have already shown that the ERG gene fusions, the ERG rearrangements can be found in a very small percentage of actually high-grade PIN lesions. It has been shown that. But we haven't investigated for the presence of the BRAF rearrangements in the early PIN lesions. But that's why we have this molecular classification. One series of actually diagnostic tests one can approach, like starting, first look at the highly prevalent aberration for the ERG, whether it is the ASAP samples or the high-grade PIN or the typical cancer samples. So I think when we do the systematic profiling of all levels of samples, we will be able to have a good understanding about what are the molecular aberrations in there.
And Jan, you might predict.
Yes, I might comment to that as well. I agree that the ERG is a very early event. It's present now. We know it's present. That it's in PINs in general. It's present in PIN, which is close to prostate cancer. That is also ERG positive, but it's in that PIN. So that's an early one. I don't know about P10, but maybe Gerhard knows about P10 in PIN lesions. He can comment on that. We didn't study that in detail. I think it's quite early, but I'm not sure whether it's in PIN or not.
Yeah, we've seen it in PIN as well using FISH. And the suggestions are probably is an early event. And I say that because when we compare patient MASH samples, we see concordance between ERG and advanced cases and early cases, and we're seeing similar concordance for P10. In contrast to AR gain, for example, which is present in later stage, but we never see it in the therapy-naive tissue.
Well, maybe you have to be a little bit careful then, because these are in situ hybridization studies, and it's not always easy to discriminate PIN from cancer or from normal cells. So there now is there might be some problems with that. But for ERG now, for instance, we have this new antibody. And the antibody really proves it's in PIN. So, and that's not easy for the P10, because it's only loss of one allele then. There's hardly induction of expression of phospho-AKT. It's hardly visible, and it's not an easy staining. And the loss of one copy of P10 diminishes P10, but again, that's not enough to be very reliable. So I think that's still open for some question, but it might be an early event, the P10 inactivation.
It's a question to Professor Palanisamy. I wanted to know, if you use sorafenib in prostate cancer, and especially in metastatic CRPC patient, sometime, this drug doesn't seem to be so active, but, it's not, based on we don't know so far how to select patient, So, and sometimes, patients have very good response, tumor shrinkage, bone recalcification, and PSA rise. How can you explain at a molecular level how PSA rise, very, very high, having a good response to this compound?
Right. So right now, I think this is in the very early investigations. We just, discovered the BRAF involvement. I don't know whether BRAF itself alone can actually cause this prostate cancer, whether it is actually cooperating with other mutations. So I think we need to investigate further to answer your questions, how well we can treat these patients,
I guess that could be really interesting if the promoter of RAF was not androgen dependent in that patient.
Right.
So, and that's clearly one of the limitations of using PSA as a surrogate. That would be a classic example.
Let's pick up PSA as a surrogate, because Gerhard, your talk on the circulating tumour cells was fascinating. The more we learn about it, and clearly there's relevance within the remit of trials, but the more we look at it, then the more we step back and say, well, it is kind of non-specific in that at one point you reopened the door from narrowing in on the CTC cell to potentially including the white cells, and you still find significant readouts in terms of response or survival. And I guess one question is that when we treat patients and we see a response with CTCs, Are you seeing a discordance then with PSA? So the CTC is going one way and the PSA is not saying the same thing?
We have seen that. So, and from 79 patients treated with abiraterone, only 4 had a decline in PSA, but no decline in CTC, an actual rise in CTC. And clinically they were progressing. But these are small numbers, and those are the sorts of questions we will be exploring.
And are they progressing with new lesions? So are the CTC seeding new lesions? And that may be the significance of the circulating cells?
But we don't know if they're seeding new lesions or it's just a readout of active metastasis.
Yeah. Just a quick question up there.
That was precisely what my question was. So there have been 2 analyses from randomized trials, one by myself and the other by Dr. Armstrong, looking at a 30% PSA decline as a surrogate marker for cytotoxic therapy. In your studies with abiraterone, are you planning on doing a comparative analysis to see whether this is actually truly better than PSA or not?
Definitely. That would be an obvious investigation.
I just have a purely speculative question, and I guess Dr. Palanisamy, this would be most appropriate for, but we have 2 preventative agents that have been studied in huge multi-million-dollar studies, dutasteride and finasteride, and I would just wonder if you could speculate if you thought these might be better preventive agents for the ETS family rearrangement tumors than the non-ETS family rearrangement tumors, and if you're looking at that in any prospective way.
Yeah. So I think for the deuterosteroid, we are actually doing a collaborative work with the GSK. So there is a clinical trial that is going on, what is called the reduce and the reading trial. So in fact, I'm the principal investigator on that. I'm working on those samples.
Great. We
are looking at the EDS profiling because it has not been done before. All those samples, we are profiling the EDS status, and I think soon we will have the results published. We have some interesting observation there.
Perhaps a comment that, yes, we see a very clear pathway to a molecular target around the gene fusion, but it still is a small proportion of patients. And where should we be going with, for example, sequencing for P10 mutations? I raise this because Cancer Research UK is initiating a collection whereby routine clinical samples will be sequenced, looking for common molecular aberrations that would potentially tailor therapies. And this is going to be a problem if we're looking for the gene fusion in a proportion of patients, and if we're looking to the P10 mutations that might give us an indication or a direction towards a PARP inhibitor. So what would you, if you were to nominate your candidate for CRUK to say, in prostate cancer, the most important sequencing that you must do, or the most important genetic aberration you must do on patients who've got prostate cancer in the UK is? Could I ask you to nominate?
But maybe you have to define what's most important then.
Yeah.
Well, I think this is towards therapeutic readout. So this is in a search for candidate therapies. And if we are looking for...
But yeah, that should show the importance is there is a therapy. Or is it the frequency or the prognosis? What?
I think these will be predictive. So will a patient respond to a therapy? Is a question that we must be asking if we're looking towards tailored therapeutic strategies. And we know, or how certain are we that we will have a response to a therapy if it's a gene fusion? And how certain will we be that there will be a response to a therapy if there is a P10 mutation?
I think it's difficult to decide. I'm not a clinician, but I think what happened so far is if you have a group of patients and you start with a trial and there are some patients that respond, and later on you're going to find out what is the genomic way it could, the way it could happen that it responded or not. For instance, is there a P10 mutation or not? And maybe you should discuss, should you do that in front or not? And probably what you should do, and that's also a comment on the first question, I think a couple of years from now, you sequence all those patients, you have the expression profile of the genes of those patients, you have the histology, and that determines what kind of therapy you will do. And that will be, well, depends on the amount of money that's available then, but that can happen very soon.
I mean, if I can make a comment, I guess the classical example is imatinib or Gleevec, which was considered a therapeutic for what was thought to be a rare indication. Clearly, if we subcategorize the disease into smaller subgroups, but develop drugs which are exquisitely effective, there's not just commercial benefits, but patients will be benefiting significantly. We studied P10 in patients treated with abiraterone, and we used FISH, and we did not see an association between genomic loss of P10 and resistance to abiraterone or sensitivity to abiraterone. So clearly, the story with P10, as Jan has said, is much more complicated.
I shall bring this session to a close by thanking our speakers for what is an educational, fantastic tour of what is likely to come in our direction in prostate cancer. Gentlemen, thank you very much indeed.